This is podcast number 27. All right, moving along in the molecular part of this class, we have been discussing how to duplicate your DNA, and now we're going to talk about transcription, which is the other part of the central dogma, turning that DNA into RNA, and then after this we'll talk about translation. So we're going to break transcription down into binding phase, the initiation phase, the elongation phase, and the termination phase. And I want to warn you that you're going to see lots of diagrams like this. They look very similar to the diagrams that you saw for DNA duplication, but this is not DNA duplication. This is making RNA from DNA. There are important differences, and I'll point those out along the way. But since your book is using one color scheme and one artist or graphics design company, they're using similar shapes and colors, and that can be sort of confusing. Okay, so we have a double-stranded piece of DNA. That double-stranded piece of DNA is going to be transcribed into RNA, shown in red here. That is going to be done by RNA polymerase. It looks similar to the diagram you saw for DNA polymerase, but this is RNA polymerase making RNA from DNA. And RNA polymerase makes RNA from DNA without a promoter. Okay, so in duplicating DNA, you saw first that you lay down an RNA primer. That does not happen in RNA transcription. You just make RNA with no primer. Okay, so that's a big difference. There is a part of the DNA called a promoter, and that's what RNA polymerase looks for to recognize as to where do I start. The promoter is considered most of the time upstream of the start of transcription. So upstream means in the 5' direction. So if the gene I'm going to transcribe is right here, follow my cursor arrow here, then upstream of that would be in the 5' direction according to this sense strand. And this light green area would be considered the promoter where the RNA polymerase is going to bind. Okay? Usually, the promoter does not get translated, does not get turned into RNA. And here they show that part of the promoter is translated. That is not usually the case. Okay, that's just an artist rendering here. Okay, you're going to see in subsequent diagrams that this is not usually the case. Okay. So usually the promoter is where the RNA polymerase is going to bind. It's what the RNA polymerase is looking for to recognize where do I start. And then RNA is made without a primer. And usually the promoter is not transcribed. Okay, and then at the end, there will be some sort of termination signal, and we'll talk about how RNA transcription stops. So, having said all that, usually the promoter is upstream of the gene that's going to be transcribed, but not always. It can be downstream. I'll give you an example of that. And we're only going to talk about examples of you have one promoter and one gene being transcribed. One gene can have multiple promoters in eukaryotes. And we're not going to talk about that case, but I want you to know that it can happen. So to talk about promoters first and talk about transcription first, we're first going to talk about transcription in bacteria, in prokaryotes. It is better studied and simpler than it is in eukaryotes. And so it's a good place to start. So if we look at a bacterial promoter and a numbering scheme, the numbers correspond to nucleotides. So if we wanted to talk about a particular feature like this TATAAT sequence, we would say that is minus 10 of the start site. So the first nucleotide that gets transcribed is +1. If I count back 10 nucleotides, this T would be minus 10. There is no 0. So it goes minus 10 down to -1, and then the first nucleotide that gets transcribed is +1, and so on. So from the origin of transcription, plus is in the 3' direction and minus is in the 5' direction. Okay, plus is downstream, minus is upstream. Okay, and again, there is no 0. It goes from minus 35 in this case to -1, and then plus, and so on. It can go much further than minus 35 and much further than +1, but that's the numbering scheme. Okay, if you think of a number line without a 0, like in number theory, it works like that. These sequences here are consensus sequences in that if we compare the sequence of many bacterial promoters, we see this sequence in common, but not always. Okay, some of these T's can be switched out for G, and so on. So they are recognized by bioinformatics programs that would say, we're looking for a sequence that looks something like this in about 10 base pairs back from an AUG, for example. And what does it look like? And if you have something that looks something like this, okay, that is likely the -10 sequence, and this is, and then 35 base pairs back, that's likely the -35 sequence, but it might not be exactly that sequence. And again, this is bacterial promoters to start. Okay, again, with bacterial promoters, the RNA polymerase has many different subunits to it. This is a more realistic picture of an RNA polymerase. One of the subunits is the sigma subunit here, and that sigma subunit has a part that's going to bind to the -10 and a part that's going to bind to the -35. That then it also has an alpha subunit that's going to bind to this upstream element. You can have transcription without this alpha subunit, but it's much more efficient and much more successful with this alpha subunit. These parts are taken off the rest of the diagrams just to make the diagrams cleaner, but they're still there. Once sigma binds to the -35 and -10 elements, it will the polymerase will pull on the DNA, opening up a big RNA transcription bubble, and it'll start to transcribe RNAs. Most of these will fail at around 8 or less base pairs. Once it gets one that is bigger than 9 base pairs, it will keep going. It'll release the sigma subunit, and then it'll keep transcribing down the DNA, reading the DNA, making an RNA transcript. So you'll have several false starts that make these short RNA practice RNAs until you get one that is long enough, and then once you have one that is long enough, it'll take off. And again, this is a bacterial promoter initiation of transcription situation. The RNA polymerase, again, they're not showing you the alpha subunit here or the sigma subunit, but just looking at the bulk of the RNA polymerase, it's going to read off the light blue. Follow my cursor here as I trace the light blue, which strand, which is the template strand, which runs from 3' on the left to 5' on the right. It's going to use that as a template. That's why it's called the template strand, or it's called the antisense strand. The 5' end of the mRNA that it transcribes will be on the left, 3' on the right, and this will become the mRNA. Okay. The RNA polymerase also has 3' to 5' proofreading ability. It's not as robust as the proofreading ability in a DNA polymerase. If you think about how long an mRNA typically lasts, it can last seconds to minutes typically, so that if there's a mistake in that mRNA, it's not as big a deal as if there's a mistake in your DNA, which you're going to replicate and pass on to future generations. So it does have proofreading ability, but it's not as robust. And again, RNA polymerase does not need a primer. DNA polymerase does, but RNA polymerase does not. It just recognizes the promoter to get started. To stop, to terminate transcription, you have in some bacterial genes, you have a region of the DNA that will transcribe a GC-rich region in the RNA, which will form a hairpin loop, this sort of secondary structure, and that structure is enough to disrupt the mRNA that is being transcribed and make it come off the polymerase DNA complex. In other bacterial genes, there is a protein called Rho, which is a termination factor, and they use that to terminate transcription. So we had initiation of transcription here, elongation, and then termination of transcription. Again, this is all bacterial that we're considering first. And here's an H5P question for you. Moving on to eukaryotic promoters and eukaryotic transcription, which is much more complicated. In eukaryotic transcription, you have not just one RNA polymerase, like you see in bacteria, but instead you have several RNA polymerases, and these RNA polymerases are specialized for making different kinds of RNA. RNA polymerase 1 makes many of the RNA parts of a ribosome, the 28S subunit, the 18S subunits of ribosomal RNA, so the RNA parts of ribosomes. RNA polymerase 3 also makes part of the RNA part of ribosomes. And in addition to some tRNAs and regulatory RNAs, RNA polymerase 2 is the one we're going to be talking about most. RNA polymerase 2 is the one that makes RNA that's going to be translated into protein, so messenger RNA. It also makes some regulatory RNAs that are a big deal in medicine now and in cell physiology now, but we're not going to talk about them more in this class. So you have three kinds of RNA polymerase that are transcribing nuclear DNA. That nuclear DNA turns into a ribosomal RNA. It's most likely RNA polymerase 1, but it could be RNA polymerase 3 if it's making one of the small subunits of ribosomal RNA. If it's making a tRNA, that's RNA polymerase 3. If it's making a regulatory RNA, that could be 3 or 2. If it's making mRNA, it's definitely 2, and we're going to talk mostly about 2. These three polymerases differ in their sensitivity to a poison called alpha-mantinin. Alpha-mantinin is synthesized by the death cap mushroom, and it poisons polymerases, specifically RNA polymerase 2. You are constantly transcribing your genes, and you need to do so for life. And this is a very toxic poison. If you just touch one of these mushrooms, that can be enough to cause death. The former professor who taught this class, Dr. Ott, in addition to being an excellent cell biologist, was an accomplished mycologist, a person who studied fungus, and he could identify mushrooms in the field, which is a very tricky thing. He was called in often to consult on cases in the emergency room where someone had been picking mushrooms and they were showing symptoms. He told us a story of consulting on a emergency room case where a whole basket of death cap mushrooms was collected by this family, and unfortunately there was nothing to do for the family and they perished. So these grow in Northeast Ohio. These could grow in your backyard. If you see anything that looks like this, leave alone. In general, leave mushrooms alone. Don't let your dogs eat them. Don't let your kids play with them unless you know what you're doing. Okay. So moving on to a eukaryotic promoter and the structure of a eukaryotic promoter, they're much more diverse than prokaryotic promoters, and their structure is more complicated. So we're going to be talking about the core promoter and an upstream control element. The core promoter is where most of these protein elements are going to bind before the polymerase can bind. The upstream control element can affect the activity of the core promoter. How likely is that promoter going to be activated? How likely is that gene going to be transcribed? It is not the same as an enhancer, which is another thing you see in eukaryotic genes, but that an enhancer tends to be very far away, like minus 50,000 in terms of its location. The same sort of numbering scheme in that the first nucleotide that is transcribed is +1, and 5' of that are minus, 3' of that are plus, and there is no 0. So in this case, -80 to -1, and then you switch over to +1. +1 is the first nucleotide transcribed. In eukaryotic promoters, again, like I said, they're very diverse. We're going to talk about RNA polymerase II promoters mostly, and that's what this slide is about RNA polymerase II promoters. The structure of RNA polymerase II promoters tends to have an initiator region, okay, and then a binding recognition element, BRE, where transcription factor 2B is going to bind, or a downstream promoter element, DPE, where a different type of promoter element, recognition element, would bind. The types of promoters for RNA polymerase II tend to be BRE plus TATA or DPE plus TATA. Both of them contain INR parts of the promoter. What I want you to know is that eukaryotic promoters are diverse, and there are different types of promoters in eukaryotic promoters. They have recognition sequences where we can recognize, okay, this is a DPE binding site for a DPE plus INR plus TATA box promoter, whereas this is a BRE binding site recognition for a BRE plus TATA box plus INR promoter. And going forward, we're going to talk mostly about a core promoter element and how transcription gets started for an RNA polymerase II type of promoter. The TATA box binding site is also seen in archaeans, and archaeans, that's part of the evidence for why we think archaeans are closer to eukaryotes than they are to prokaryotes that don't have this sort of structure in their promoters. Okay. Just to show you a different type of promoter, this is one for RNA polymerase III. This is binding to transcribe tRNA, for example. They can have promoters that are downstream of the start site. And we're going to talk mostly about situations where the promoter is upstream 5' of the start site. Here's an example of where it's downstream, but this is RNA polymerase 3, and we're going to talk mostly about RNA polymerase 2, which is making mRNA that will be translated into protein. All right. I'm going to go through this diagram a couple times. This is a diagram I would like you to know in detail and be able to describe in detail. This is talking about general transcription factors. So for all kinds of different promoters, this one is going to talk mostly about RNA polymerase 2. There are proteins that need to be laid down in a certain order on the DNA in order for transcription to get started. Those proteins are called transcription factors, and they are not specific to a particular gene. No matter what kind of gene you're going to turn into mRNA, these things have to be laid down first. They are not the only thing that needs to be laid down. There are specific transcription factors that may be specific for a certain kind of gene. So transcription factors that may be important for fat metabolism genes or for glycolysis genes, for example. But this happens regardless of the kind of gene it is, and that's why they're called general transcription factors. They all have these wonderful names. There's just a string of letters and numbers. To decode these, like TF2D. TF stands for transcription factor. The Roman numeral 2 refers to RNA polymerase 2, and D is the specific type of transcription factor. So this is a protein that binds to a core promoter that is for RNA polymerase 2, and it is the specific protein D. Okay. Transcription factor 2D binds to the core promoter first, followed by 2A and 2B. Then 2F, along with RNA polymerase 2, binds to the core promoter. So this is the thing, RNA polymerase 2 is the thing that's going to make mRNA. And note that it doesn't even come into the picture until 2D, A, B, and F come into the picture first. Okay? In this diagram, you'll see that the RNA polymerase has a C-terminal domain tail. So all proteins have an amino terminal where the first amino acid is, the N-terminal. And then they have a C-terminal where the last amino acid is. And this is at that end of the protein where there are several amino acids that form a tail. That'll become important in a second. Then, after all these get laid down, 2E and 2H get laid down. 2H has helicase activity, which allows it to unwind DNA. It also has kinase activity, which will phosphorylate the protein. That's what kinases do. The tail gets hyperphosphorylated by 2H, and then that hyperphosphorylation event allows the tail to leave the promoter and start transcribing that gene. So let's go over that again. First, transcription factor 2D binds, then A, then B, then F, along with RNA polymerase 2, joins the complex, then E, and then H. And H is the one with the helicase activity, which is going to allow the DNA to unwind, and then also has kinase activity, which hyperphosphorylates the C-terminal domain of RNA polymerase, and that signal allows the RNA polymerase 2 to leave the station and start transcribing the DNA. So you can think of this phosphorylation is sort of the gang's all here, in that until you get this complex forming, H is the last thing to join, and H is the thing that's going to phosphorylate this. So H is not going to join in unless E, F, A, B, and D are already there. And so that is that quote unquote tells the RNA polymerase that the assembly is complete and you can start transcription. And this is going to happen regardless of what kind of protein coding gene it is, and there will be more transcription factors depending on the type of gene it is. So I am likely going to ask you to do a well-labeled diagram with text, so it would be good to practice that last sequence that's on that slide. Here is an H5P question for you. This TATA binding protein, which we skipped over, but it's right there, TBP, part of TF2D, TBP, right there. This thing is a protein that binds to the minor groove of DNA. When it binds to the minor groove of the DNA, it puts a kink in the DNA, bends the DNA, and it's an important part of the core promoter protein binding sequence. It's involved in all type of eukaryotic promoters, and so it's an important part of setting up transcription.